Assalamu alaikum wa rahmatullahi wa barakatuh. Good day, everyone. Welcome back. This is National Science and Mathematics Olympiad, NSMO. And don't forget, our goal is analytical thinking. Olympiad questions, of course, is not easy question. It's not that simple questions. So we are focusing not just memorize, no, it's just to dig deep behind the information. So stay tuned. This lecture is part of phase 1, which focuses on understanding why and how meiosis happens, not just memorizing facts. Mitosis creates identical cells for growth. Meiosis creates gametes, sperm and egg cells. And we're still here in chapter 4, cell division. This is your instructor, Muhammad Al-Ali. I welcome you back. In the previous lecture, we learned mitosis and cell regulation. Today, we answer, why do we need a different divisions method? Think about it. Why? Because reproduction requires half the chromosome number. So it's another story to tell. So that's what we are taking today, inshallah. Welcome to our fourth lecture. Today we explore meiosis, a different form of cell division that creates diversity. Unlike mitosis, meiosis doesn't produce identical copies. It produces uniquely different cells. In this lecture, we will understand what meiosis is, why chromosome number must be reduced, and what makes its stages unique. Pay attention to chromosome movement. This is key for problem solving. Don't forget that. Let's start with meiosis, talking about characteristics of meiosis. And let's look at this diagram. This diagram represents meiosis where homologous chromosomes, separate, resulting in 4 cells, each cells containing only one set of chromosomes. Also, if you see here, there is 2 colors, the red and blue. So the different colors, red and blue, show that each chromosomes comes from one parent. So probably the red one come from father or mother, blue one come from father, mother, or father. It doesn't matter, but different gender. Okay, and during division, they are randomly distributing among the gametes. So probably here, 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 sometimes come here, here, randomly. It's not like a rule. And the result is genetic. It's not like mitosis, identical. Here, the difference in genetic variation, because they produce cells. Because what? Because each cells receive different combination of chromosomes. So this is the purpose of meiosis. So let's answer these questions, upcoming questions. Where is it occurs? Of course, it occurs in gametes. 6 cells. Okay, the number of resulting cells, as you see, genetically different for 4 different cells. That's it, 4 cells. And the number of chromosomes, what the number of chromosomes? The number of the chromosomes is half the chromosome number. Half the chromosome number, and we called it N. N. It's mean to be from father or mother. And example for it, it's in egg or sperm. Egg cells, pollen or pins. Okay, so remember this. Without this reduction of number of chromosomes, the resulting zygote would contain double the chromosomes, distributing life processes. So if there's not reducing, there will not be shape of life. Do you understand it now? Let's move on to another important term in meiosis, which is crossing over. So, crossing over is a process in which segments of chromatids are exchanged between two homologous chromosomes. Look to this diagram, guys. This diagram shows the process of genetics crossing over, where homologous chromosomes come close together during the first meiotic division and exchange segments of their chromatids, resulting in a new chromosome with a unique genetic combination different from both parents, as we see it here as a result. Okay, so this exchange of genetic material is a key reason behind the variation of traits among individually of the some species. Imagine, let me give you this statement. Imagine each chromosome carries information from the father or mother during crossing over. This information gets mixed, okay, creating a new and unpredictable trait combination. That's why crossing over is really, really important in meiosis. Before watching this clip, have you ever stopped and asked yourself, why do brothers and sisters share features yet never look exactly the same? Why are no two humans on Earth identically except twins? In this clip, we dive into one of nature's most brilliant secrets, meiosis, the process that creates life endless diversity. It doesn't just divide its cells, it mixes genetics like a master artist, blending colors, creating combinations that world has never seen before. Meiosis is the reason species evolve, populations survive, and you are who you are, uniquely designed, unlike anyone, past, present, or future. So get ready. The story of how life reshuffles its deck begins right now.
In this lesson, we'll explore the details of what happens during the phases of meiosis. Meiosis, sometimes called reduction division, is the type of cell division that produces gametes. By gametes, we mean sex cells, such as sperm cells in males and egg cells in females. Meiosis is broken down into two stages of cell division, called meiosis I and meiosis II. Meiosis I has four phases: prophase I, metaphase I, anaphase I, and telophase I. And meiosis II also has four phases: prophase II, metaphase II, anaphase II, and telophase II. Let's look at what happens during meiosis I. Prophase I starts with a diploid cell. Its chromatin contains two uncoiled, spread-out sets of chromosomes, one from each parent. After the DNA in the chromatin replicates, it condenses into the more familiar X-shaped chromosomes. The replicated DNA is the same in the identical sister chromatids of each chromosome. In a process called synapsis, each chromosome pairs up with and binds to its corresponding homologous chromosome, forming a tetrad. A tetrad is the group of four sister chromatids in paired homologous chromosomes. The chromosomes contain genetic information called genes. These genes were inherited from each parent. And different versions of the same gene on each chromosome are called alleles. In a process called crossing over, chromatids from each homologous chromosome exchange segments of alleles. Also called recombination, crossing over randomly happens on every chromosome, resulting in different gene combinations. This explains why every gamete is genetically different from every other gamete. Crossing over results in genetic variety in offspring. This is why children are different from their biological parents, as well as from their biological siblings. Continuing on with prophase 1, the nuclear membrane disappears. The centrioles move to opposite ends of the cell, and spindle fibers fan out from them. Next, in metaphase 1, the homologous chromosomes line up at the equator and attach to spindle fibers from opposite poles. During anaphase 1, spindle fibers separate the homologous chromosomes in each tetrad and pull them to opposite poles of the cell. The cell enters telophase 1 with one chromosome from each homologous pair at separate poles. However, each chromosome still consists of sister chromatids. Keep in mind that each chromosome's sister chromatids are no longer identical because of the allele exchange that happened during crossing over. Then, spindle fibers disappear and the nuclear membrane reforms around the chromosomes. Finally, cytokinesis occurs. Meiosis 1 ends with two genetically different haploid daughter cells. Each haploid cell contains only one set of chromosomes consisting of paired sister chromatids. Both cells now enter the next stage, meiosis 2. However, unlike meiosis 1, DNA does not replicate before meiosis 2 begins. Once again, in prophase 2, the nuclear membrane disappears, and spindle fibers fan out from the two sets of paired centrioles. During metaphase 2, the chromosomes in each cell line up at the equator and attach to spindle fibers from both poles. During anaphase 2, the sister chromatids of each chromosome separate and move to opposite poles. Once the sister chromatids separate, they're called chromosomes. Finally, during telophase 2, the spindle fibers disappear and nuclear membranes reform. And cytokinesis occurs in both cells. Meiosis 2 ends with 4 genetically different haploid daughter cells, each containing only.
Let's examine ourselves if we are understanding meiosis very well with this exercise. In the following graph, to which stage of meiosis does number 2 correspond? That number 2. And before I fulfill the options, let's illustrate this figure. Okay. The horizontal axis shows the time, while the vertical axis represents amount of DNA in the cell. At point 1 here, the DNA level is constant, indicating the cell is before replication. Okay, here, so before replication, the DNA. While about point 2 here, the graph shows increase from 2C to 4C, which means DNA replication is taking place. At point 3, DNA remains high, showing preparation before the first meiotic divisions. Okay. Point 4 here, DNA decreases by half, which is after meiosis 1. And at point 5 here, DNA drops again after meiosis 2, forming haploid cells. Okay, let's have the options now, and let us illustrate them together. G1. Why G1 is wrong? Because DNA does not replicate here. The amount stays constant. The match is 1, not 2. Okay, what about B? So G2. In G2, the DNA is already doubled, but not increasing. This is stage 3, not 2. So also that one is wrong. What about S-phase DNA? Here, DNA replicated increase from 2C to 4C, matching point 2. So this is the right answer. But let's keep going with D. Meiosis 1. DNA here decreases in this stage, matching 4, not 2. So this is also wrong. So the right answer is C. S phases DNA synthesis. Do you get it now? Let's move on to another exercise. If the number of chromosomes in a sex cells before meiosis is 32, how many chromosomes are there in anaphase 1? Is it 8 or 16 or 32 or 64? What do you think? So the concept here, in anaphase 1, homologous chromosomes separate, not chromatids. That's the main concept. So this means that the chromosomes number does not decrease during anaphase 1. The reduction happens only after completion of meiosis 1, when cells split. So the number remains 32 chromosomes during anaphase 1. So the right answer would be C. Do you get it, guys? So why? Because in A, 8, this is too low, so this is wrong. Okay. And in B, which is 16, this is the reductions happens after meiosis 1. So also that one we didn't need it. We said C, 32 is the correct one. D, 64, this is wrong. Why? Because this would require duplication. Okay. And the image here shows homologous chromosomes pairs colored differently. They are begin pulled apart as pairs, not as chromatids. Do you understand? And this is confirmed the stage is anaphase 1, where chromosomes number remains the same. This is the tricky one. Do you get it? Moving to the last exercise. Crossing over occurs during prophase 1 or prophase 2 or interphase or prophase of mitosis. Okay, the main concept here, crossing over is the exchange of genetic material between homologous, and it happens only when homologous chromosomes pair tightly. So this pairing happens during prophase 1 of meiosis. So here, prophase 1 is the right answer. Prophase 2, why it's wrong? Because no homologous pairs present here in prophase 2. What about interphase? Why it's wrong? Because interphase is only DNA replication. And why prophase of mitosis wrong? Because chromosomes do not pair here. Okay, and this image, it shows two homologous chromosomes crossing and exchanging segments, and different colors indicate the genetic exchange. And this process creates a new combination resulting in genetic variation. Okay, so be notice, crossing over only happens one in prophase 1 and never repeats in prophase 2. Okay, save this in your mind. Finally, let's recap what we have learned so far. Today we learned that meiosis reduces chromosome number and recombines genetic information, explaining biological diversity. Thank you for your focus. Think about this. How can small errors in these stages lead to genetics disorders? Assalamu alaikum wa rahmatullahi wa barakatuh. See you soon.